of the conference. So we will start the session, today's session, the morning session, with the first talk by Dr. Ayyasamy Vanniarajan from the Department of Molecular Genetics, Aravind Medical Research Foundation from India. So the title of the talk is Emerging Role of Tumor Microenvironment Derived Exosome in Therapeutic Resistance and Metastasis through Epithelial-to-mesenchymal Transition. So Dr. Vanniarajan, the stage is yours.
Thank you very much, Christiana and Ira, for the opportunity given. I'm really honored to be a part of this meeting, and I really miss to be to see all of you in person. But this is a good platform to talk our research and to interact. So thank you again for the organizers. So I am from Aravind Medical Research Foundation. It's a research wing of Aravind Eye Hospital, Madurai. So it's a southern part of India, and it is close to the tip of southern India. So what I'm going to talk on next 25 to 30 minutes is about the eye cancers and how we see these EMT and the exosome derived EMT is a part of the transformation in our cases. So this is a perspective article published recently. So Ira saw this article and asked us to give a presentation. So with our recent data, I'm going to talk on it. Basically, the eye cancer, what we deal here in India is a threatening disease. So that is a case that can cause the damage to the eye, sight, and as well as the life in many cases. Because these cases are presented as very late, and the diagnosis is also a problem, and also the treatment is also a major challenge. So every year about 10,000 new eye cancer cases are diagnosed in India, of which 70 to 80% are in adults and 20 to 30% in children. So about 1,500 children are diagnosed with retinoblastoma, which is the most common intraocular tumor, which I'm going to talk in detail at the end of it. And the other cancers include lymphoma, adenoid cystic carcinoma, squamous cell carcinoma, and melanoma. So what are the major challenges of these eye cancers? As like any other cancer, the course of the disease is a major enigma for the clinicians, because each disease remains unique and their course of progression is quite challenging. And some of them are quite responsive to the chemotherapy or radiotherapeutic drugs, whereas it still remains as a highly variable among the patients due to their individual genetic setup. And another major problem in the treatment is the tumor recurrence. So many of the patients, they get cured and they go back to home and they don't come for further follow-up. And all of a sudden they come with a major recurrence. So this again is a major challenge for our treatment, treating clinicians. And another major problem is the metastasis disease, which I'm going to talk, and this chemo resistance I'm going to talk about in detail today. So in order to understand that, first we need to understand the disease first. So in any cancer, there are major hallmarks. First, the limitless potential to replication, and then the sustained angiogenesis, where they get feed from their neighboring cells and evading apoptosis, and also they get self-sufficient growth signals and insensitive to the antigens, finally leading to metastasis to the next organs due to invasion and metastasis. So the cancer cells alone cannot cause all this. So they convert the neighboring cells in the tumor milieu, which is called the tumor microenvironment. The tumor microenvironment constitutes the entire portion of the tumor milieu, which includes the tumor cells and the neighboring stromal cells. It also encompasses the extracellular matrix along with the blood vessels that helps in angiogenesis and also metastasis invasion. These tumor microenvironment, they also secrete major, also part of the macromolecules such as proteins, lipids, carbohydrates and nuclei that are synthesized by these cells. All these components, they orchestrate a variety of tumorigenic through a process called epithelial-to-mesenchymal transition called EMT. So these can execute the functions such as the chemoresistance, metastasis, angiogenesis, making the immune privileged environment of the tumor milieu. So how this tumor microenvironment prevents, helps in chemoresistance is, they prevent the diffusion of the chemotherapeutic drugs by raising the interstitial fluid pressure of the ECM and further reduces the drug bioavailability to the site of the tumor. The cancer stem cells are the major commanding cells. So they regulate the aggressive phenotypes with the help of the stromal population inside the TME. So any containment of the tumor growth and the progression should definitely consider the modulation of the TME, not only with the cancer cells, but along with the TME. So what are the components of this tumor microenvironment? So the tumor microenvironment majorly compasses by the cancer stem cells and non-cancer stem cells, but distinguishing between them is also challenging. These cancer stem cells are highly tumorigenic with having the phenotypes of self-permeability and quiescence, whereas the non-cancer stem cells, they are poorly tumorigenic cells and they are differentiated. Along with that, there are various other cells that can get transformed, mainly the fibroblasts that can get transformed into a cancer-associated fibroblast. These are the predominant cell type that are involved in the synthesis and remodeling of ECM during the wound healing. And the mesenchymal stem cells that are the multipotent stromal cells, which are induced by the cancer cells to differentiate into cell lineages such as CFs, macrophages, and endothelial cells. These endothelial cells, apart from that, mesenchymal stem cells are also inevitable components of TMB because they initiate the angiogenesis from which the tumor cells get nourishment and also get oxygenated. The nascent blood vessels spawned with angiogenesis can also initiate metastasis. Apart from that, adipocytes can also promote metastasis in invasion. Along with that, the immune cells that also play a major role. The CSCs convert these macrophages, neutrophils, and naive T cells into tumor-associated cells. The CSCs also neutralize the anti-tumor immune components such as NK cells, cytotoxic T cells, and other immune cells. So this is very intricate, whereas the cell-to-cell communication first starts among the heterogeneous cancer cells that triggers the EMT. The cancer cells then induce the trans-differentiation of the fibroblasts into CAF, and then the cancer cells induce the differentiation of the mesenchymal stem cells into a CAF or other tumor-associated mesenchymal stem cells. Finally, these TAFs and cancer mesenchymal stem cells, they play a major role in the de-differentiation of the cancer cells into cancer stem cells, resulting in EMT and chemoresistance. These cancer cells also enhance neovascularization of the endothelial cells, leading to metastasis, and these cancer stem cells communicate with the cells such as macrophages, neutrophils, naive T cells, making the environment into immune privileged TME. So all these intricate TME are facilitated by the exosomes. The exosomes are the in extracellular vesicles with a diameter of 3 to 150 nanometer that sets off from the cells into the ECM, and they share the phenotype of their parental cells. The exosomes, they are ubiquitously present in any body cells such as blood, saliva, urine, amniotic fluids, and many other body fluids, and thereby it serves as an inevitable biomarkers in disease prognosis. They carry various components, including nucleic acids and also many other proteins. So they have various ceramide that helps the exosomes to serve as signalosomes, and they have the phelotillin, which regulates the cargo sorting and trafficking. The actin filaments also assist in the exosome movement. The exosome on the surface have the CD63 and CD81, which serves as the exosome markers. These exosomes, they also help or they are derived from the endosomal root based on the unique properties, and they deliver various biomolecules precisely through receptor-mediated trafficking and tumor progression. The exosomes-mediated intracellular trafficking is an evolutionary adapted mechanism, as these exosomes are resistant to a variety of harsh, enzymatic, degrading, and hypoxic conditions. So thereby they thrive on the TME of the leading to aggressive disease. So these exosomes play a major role in the tumor microenvironment in multiple ways. The equilibrium of the CSCs and non-CSCs can be maintained in aggressive malignancies by either dedifferentiation or enrichment of the CSCs under pressurized conditions such as chemotherapy. The chemotherapy can induce certain molecular changes in TME. The cancer cells may evolve to produce the exosomes that are bound in microRNA to tolerate these adverse conditions. So the various microRNA such as the microRNA 9-5P and microRNA 195-5P and microRNA 203A 3P that eventually activates the stemness-related genes such as the Notch1, SOX2, SOX9, OCT4, and NANOG by directly targeting the 1-PET1-2 transcripts in recipient cancer cells. So these molecules in turn activates the dedifferentiation of non-cancer stem cells into cancer stem cells, selecting overexpression of ABC transporters observed in the cells, suggests that transactivation of the stem cell related genes to affect the chemotherapy drugs to evade the treatment. So apart from this, the cancer stem cells also shed exosomes that facilitate the expansion of the CSC population TME. In renal cell carcinoma, the CSC derived exosomes with microRNA 119B3P activate the conversion of residual non-cancer stem cells into cancer stem cell, deriving the organotrophic metastasis. The silencing of the P10 and E-catrin is activated by this microRNA, leading to upregulation of the EMT genes N-catrin, vimentin, and also twist. The CSC derived exosome can also transfer functional Wnt3A, which I am going to show specific Wnt signaling at the end. So they also can lead to various other stromal cells such as the fibroblasts, bone marrow adipose derived mesenchymal stem cells. The conversion of the fibroblasts into CAFs is facilitated through activation of the SMAD pathway by TGF, which is abundantly present in the cancer stem cell derived exosomes. About 80% of the TGF produced by bladder cancer cells were found to be exosome bound, and when the phosphorylating SMAD by TGF receptor is found highly mediated by exosomes. Apart from this, microRNA 21-5P is also transported to fibroblasts by CSC exosomes that induce the differentiation of fibroblasts into CAFs by direct inhibition of P10 and PDC4 activation of the STAT3. Reciprocally, whatever the activated CSF can secrete exosomes that promote the tumor growth, derived exosomes that were shown to be highly active. These ultimately, these molecules, along with the various other molecules such as the mesenchymal stem cells, which induce the differentiation of the multi-lineages into macrophages and endothelial cells, that can also promote the chemoresistance and metastasis. The cancer cell derived exosomes that can activate pro-oncogenic pathways and facilitate the conversion of CSF. And this TGF beta 1 is also associated with increased secretion of pro-angiogenic metastatic proteins such as VEGF, hepatocyte growth factors, MMP1, MMP9, MMP13 by MSC derived CAF, which enhance their motility. Besides this, the breast cancer derived exosomes activate the various TGF beta and various other molecules. Apart from this, the endothelial cells are also inevitable components of the TME. They initiate the angiogenesis from which the tumor cells get nourishment. These nascent blood vessels formed by angiogenesis, they initiate the metastasis by encouraging the cancer cells to migrate. The outgroup death of tumor cells in TME implements the metabolic demands and also hypoxic atmosphere. Among the immune cells, the cytotoxic T lymphocytes and NK cells were the major components that challenge the tumor cells, and these can convert these naive T cells and neutrophils that can cancer-associated immune T cells. These can cumulatively introduce the tumor immune privileged environment The incorporation of PDL1 into exosome is one of the major factors of immunotherapeutic resistance and also poor prognosis. So there are various other molecules involved in this exosomal transfer. So mainly the downregulation of the MET1 and also the various other genes that are involved in these changes. So these molecules that thereby they further act on the CSC and non-CSCs, further promoting the metastasis through endothelial cells by the transfer of upregulation of metastasis-related genes such as ICAM1, VEGF, and various other genes that lead to naive blood vessels, leading to metastasis to the next organs. So majority of these are affected through Wnt pathway and EMT. So mainly these are activated in 4 steps: the exosomal Wnt pathway proteins, mainly the Wnt3A, and exosomal Wnt agonist. This mediated indirect activation of the Wnt pathway is also done by agonists such as activin A, UBR2, and FGF2, and exosomal factors that target this Wnt agonist. And finally, this lead to the activation of the various genes of EMT and metastasis. So taking these all together, these exosomes are generally studied by the various separation of methods such as the differential centrifugation or ultracentrifugation. And these exosomes are also precipitated through various commercially available reagents. And they are also integrated through double filtration microfluidic devices. So these are the best methods to study these exosomes, which are currently in practice. And then they are characterized by the nanoparticle tracking analysis, transmission electron microscope, and flow cytometry. So what we have done, we are optimized the technique of ultracentrifugation and the nanoparticle tracking analysis. And this we are currently in progress. So taking to our perspective on retinoblastoma, so the retinoblastoma is a highly malignant intraocular period tumor. Mostly they come with the severe phenotypes. Most of them are diagnosed at the late stages. And during our this period, study period 2013 to 18, we have diagnosed 214 new RV patients, out of which 70% of the eyes were enucleated, most of them due to the advanced disease and poor response to therapy. So these RB cases, they are mostly biallelic inactivation is seen. About in 90 to 95% of cases, we have checked using a specific strategy by a stepwise method. And we are also interested to look for the other genetic changes, not only RB1, where we found out the various genes such as E2F3 and KF14. These change alterations in these changes genes cause the tumor progression. So it is explained in a stepwise manner. So initially, the acquisition of the RB1 mutation happens, and that lead to further changes. So the RB1 initiates, there the clonal population happens, and tumor genetic instabilities happen. And this is made worse by the acquiring the phenotypes such as the chemoresistance and metastasis in these cases, clearly demonstrated in our clinical cases. So what we have done, we have developed the resistant cell lines in our population. We have studied the Y79 cell line, which is a retinoblastoma cell line. We have created the resistance lines by continuous developing a continuous method of establishing the etoposide and carboplatin continuous treatment with a periodical break of 15 days. And these cells have been shown to show resistance with the drug index of 3.98 and also 3.95 in both the cases. So these cells were further used to study their kinetics to show how they are behave in the culture. So the doubling time was significantly increased in the resistant cells compared to the naive cells. So the increase in doubling time that gave a speculation that there could be a epithelial-to-mesenchymal transition induced by the cancer stem cells. So further, we have also checked the software agar assay, where we found the normal cells does not proliferate much, and the self-renewability of these cells shown the bigger colonies, the colony size was measured and compared. So the resistant cells showed a higher colony compared to the control cells. So these confirmed that there is a EMT transition happening in these resistant cells that lead to the bigger size and further motility. So further, these cells were also checked in a transfer assay where the cells showed a clear invasion in the matrixal cells compared to the control cells, about 100 times or 100 times more cells were shown to be showing the invasion in these cells. So that this again confirms there is a EMT transition derived stem cells. So further to confirm, we study the various markers as we have shown earlier. We found the increased expression of the SOX2, NANOG and ACT4 and KLF14 overexpression of these markers in the cells that are studied at the RNA level as well as the protein level that showed a higher expression of these CSC markers, which again confirms the EMT transformation in these resistant cells, whereas the differentiated markers are downregulated. So when we looked at the patient samples, so there we found a increased tissue differentiation as the tumor progresses compared to the non-responsive RB in the tumor where there is a continuous progression. Here it's a responsive RB, whereas the non-responsive RB, there is a bigger mass as keep progressing. So these showed a whole differentiated pattern compared to these cells. So this again an indication of the cancer stem cells, which we are going to further characterize with the stem cells. So all these show a high invasion to the neighboring cells with the retrolaminar invasion, and whereas the non-responsive RVs show a very high invasion in these next neighboring cells. So further to confirm, confocal microscopic study also showed this NANOG higher expression in the responsive RB, whereas the non-responsive RB showed a higher expression compared to the non-responsive RB, which again proves evidence for the EMT in the RB chemoresistance and metastasis. So with this, we have commanding evidence that the EMT could be a potential regulator of the chemoresistance in RB with the overexpression of these various markers, SOX2, NANOG and ACT4 that play an important role in EMT of RB. So currently we have optimized the, as I mentioned, we have optimized the ultracentrifugation methods for the isolation of the exosomes, where it is the work is in progress, and nanotracking method to isolate the characterized exosomes. And we are we are studying these exosomes distinctly, how this RB microenvironment EMT in the aggressive disease with metastasis that can lead to chemoresistance by replacing the established resistant models and non-responsive clinical RB methods. Further, to illustrate the role of the exosomes and its traffic in the EMT factors, we plan to isolate these exosomes from this patient serum by ultracentrifugation and study them also by the Western blood. So thus, our cells show the EMT and role of exosomes, which we will be confirming soon with the patient tumor cells also. I would like to thank the clinical partners, Dr. Usha Kim and Dr. Kim and Shanti, who backed up this study, and also on the research side, Professor Muthukarpan and Vanniarajan and other team members of my lab. They have helped to pursue this study. Especially, I would like to thank Segaran Balaji, who just completed his PhD in this topic, and now he's planning to pursue his postdoctoral study. So Madurai is called the Athens of East. So it has a lot of monuments, and I welcome you all sometime or the other. You can, if you get a chance to visit India, and especially Madurai. Thank you. Now I can take any questions.
Thanks a lot, Dr. Vanniarajan, for very interesting presentation. I don't see question in the chat, so I would like to ask audience to post a question in the chat. Meanwhile, I will start with a couple of questions.
Yeah, sure.
So the first one is, it has been published some years ago by the group of Rogero De Maria that cancer stem cells can also differentiate, I mean, into endothelial cells, creating the neoangiogenesis associated with the tumor. I'm wondering if you have any data regarding that, and even if an exosome can mediate this process in your model or radio of tumor model of ice tumor.
Yeah, as I mentioned, we know that this has been published, but we have not attempted that in detail by segregating out the individual cells. So whatever the evidence we have got is based on the overall tissue models. So now we have to segregate them and then we have to study that in detail. Currently, we don't have any data on it.
Okay. The other question I have is moving more further in the therapeutic application of your results. How do you envision to in some way modify the exosome or some drugs that can modify the content of exosome to block the formation of cancer stem cell progression of tumor?
Right. Currently, there are many inhibitors known to like exosome secretion or biogenesis. For example, the GW4869 has been widely studied that inhibits the neutral sphingomyelinase that can catalyze the hydrolysis of the sphingomyelin ceramide. So this helps to prevent the exosome biogenesis and secretion. So similar other compounds are being developed. So these have been tested in various cells. So once we nail down that these exosomes are the major players in this, so then we can treat them with these various inhibitors like Solaran or GW4869. So that will help in inhibiting this exosomal transport, thereby considerably reducing the EMT in the TME.
Okay. Okay, so thank you. I don't see other question in the chat. So I would like to thank you for a very interesting talk.
Yeah, thank you very much, Christiana.
And so we can move to the next speaker. Thanks a lot again.
Yeah.
So next speaker is Dr. Edison Fonseca Teixeira from the University of Melbourne, Australia. And her talk his talk is entitled Small Extracellular Vesicle Amplify TGF-beta Signaling and Promote Breast Cancer Progression. So please, Dr. Teixeira, the stage is yours.
Thank you. So share my screen and to start the presentation. All right, so hopefully you all can see my first slide.
I cannot see yet, but let's see.
Now, okay, let me. I stop sharing. Let me try it again.
Yes. Okay, yes, now we can see your presentation. Thank you.
Perfect. Let me put in the presentation mode. Okay, great. So good morning, everyone. As introduced, my name is Ayyasamy Teixeira. I'm a PhD student. I appreciate the doctor title, but I'm still a PhD student. I'm in my third year, and I'm studying the University of Melbourne. And today I would like to present part of my PhD project entitled Small Extracellular Vesicles Amplified TGF-beta Signaling and Promote Breast Cancer Progression. This project is supervised by Professor Peter Tendak and Dr. Hongjian Zhu. Before I start talking specifically about my results, I would like to offer a little bit of context about the disease itself and the extracellular vesicles. So breast cancer is the most incident neoplasia in the world, but thank you to early diagnosis and also to improve treatment, we can see on the graph on the right side that most patients present a very good 5-year relative survival. The exception here is for patients diagnosed in stage 4, which represents patients diagnosed with metastasis. Metastasis is a multi-step process The epithelial-to-mesenchymal transition, or EMT, it's one of the most important because it happens very early during cancer progression towards metastasis. During EMT, these epithelial cells will lose the attachment to surrounding cells. Will also observe the degradation of the basement membrane, the reorganization of the extracellular matrix, and as the EMT progresses, we also observe the increasing TGF-beta signaling. This increasing TGF-beta signaling inducing EMT usually leads to the loss of epithelial markers such as ecadrine and ZO1, and the overexpression or the increasing in the expression of mesenchymal markers such as encadrine and vimentin. A master inducer of this process, as is TGF-beta, it's important to understand how its signaling pathway it's established and how it operates. We can observe in this slide that TGF-beta is synthesized and secreted in a latent, as a part of a latent complex, and after the deposition in the extracellular matrix, it will interact with integrins and other proteins to release the active ligand, the free ligand, the soluble ligand. This ligand can then interact and bind to surface receptors, and after ligand receptor binding, we can have the TGF-beta signaling transduced throughout the plasma membrane. This leads to the phosphorylation of the TGF-beta receptor type 1 by the TGF-beta receptor type 2, and then the activation, the phosphorylation and activation of cytosolic proteins is SMAD2 or 3. When phosphorylated SMAD2 and 3 will form a complex with SMAD4 if not inhibited by the activity of SMAD7, and once forming this complex, SMAD2 or 3 and SMAD4 will translocate, will accumulate in the nucleus to regulate the expression of target genes. Because this is a very important, very critical pathway to cancer cell EMT and metastasis, different strategies have been developed to target, inhibit the activation and sustain of TGF-beta signaling. This includes antisense oligonucleotides, antibarriers and ligand traps, and integrins, but also kinase inhibitors. It's very interesting to understand that the inhibition of TGF-beta signaling pathway in clinical models definitely impair cancer metastasis. This is very well established. Still, the translation of these pharmacological inhibitors from bench to bedside is still a problem. Most of these drugs failed in clinical trials, raising questions about our current understanding of TGF-beta biology. We have recently published a review perspective discussing many of these points that could be the remaining challenge to use these anti-TGF-beta therapies to improve the survival and the treatment of cancer patients. One of these points I would like to highlight, it's very related to our project, is the secretion of exosomes, the biogenesis and secretion of exosomes. They could operate as a mechanism for both TGF-beta secretion and also signaling amplification. Exosomes are major mediators in cell communication. They are considered small extracellular vesicles, so most of them, at least, are expected to be small extracellular vesicles with a diameter ranging between 30 and 100 nanometers. They express typical markers such as ALEX, TSG101, and also some tantruspannings. The biogenesis starts right after the endocytosis. Early endosomes will, instead of moving on to the lysosomal pathway, will maturate and form multivesicular bodies. This involves different excrete complex sorting protein cargo, sorting different types of cargo in these multivesicular bodies, and generating these multivesicular bodies with intraluminal vesicles inside. Thanks to Rab enzymes and also to snares, these multivesicular bodies can fuse to the plasma membrane, releasing intraluminal vesicles now as exosomes. These exosomes in the extracellular milieu can then interact with recipient cells, with the plasma membrane, with recipient cells through different types of molecules, including integrins and different types of carbohydrates. And after endocytosis, the cargo is then used, released, or integrated to the recipient cell. This exosome, these small extracellular vesicles, can transport very different types of molecules. This includes several proteins, DNA, RNA, lipids, carbohydrates in this composition. And what is very important for this project in particular is that TGF-beta is one of the described cargo in exosomes, and they can be secreted both by cancer cells and also by non-cancer cells. Sorry, think I have a problem with my slide. Okay. More than the TGF-beta, it's a part of these exosomes or secreted these exosomes is that this form of TGF-beta, this exosome of TGF-beta, can also activate TGF-beta signaling pathway, induce immune suppression, fibroblast activation, EMT, migration invasion, and in metastasis, leading to metastasis. And that may explain the position of these exosomes and the presence of TGF-beta inside these exosomes may be blocking the interaction of TGF-beta with parts or with specific types of therapies. May explain, for instance, why therapies such as ligand traps or TGF-beta failed in clinical trials because they simply cannot access the TGF-beta, they cannot interact with TGF-beta. And that's from a therapeutic point of view. From the biological point of view, for understand the biology of TGF-beta, the exosome secretion by itself, it's not clearly understood. And it's very well possible and speculated that will increase during cancer progression, which could lead to contribute to and lead to increasing in TGF-beta signaling pathway activation as well. So the aim of this project was to establish the role of small extracellular vesicles, again, most of them exosomes, expected to be exosomes, in TGF-beta signaling activation in breast cancer progression in vitro and in vivo. So our first step was actually to screen some highly metastatic cell lines and some polymetastatic cell lines to understand if they differed or not in terms of exosome secretion, in terms of small EV secretion. As you can observe on the left side, different cancer cell lines from different origins present more or less the same pattern. The highly metastatic cell line, in this case, for instance, MD823-1 breast cancer cell line, secrete more exosomes than the polymetastatic MCF7 cells. This is also true for the T24 bladder cancer cell line compared to RT4. At the same time that this highly metastatic cells secrete more exosomes, more small EVs, they also present increased levels of TGF-beta signaling activity or higher response to TGF-beta. And again, I will highlight the breast cancer cell lines that we have for comparison, MCF7 and MD823-1. And as you can observe, MCF7 in response to TGF-beta, if we treat these cells after 24 hours, we evaluate the SMAD3 transcription activity by luciferase assay, we observe usually something around 8 to 10-fold induction. But when we do the same with MD823-1, we can reach a fold of induction that is up to 1,000. So that's raised the questions about these exosomes. So these exosomes could be indeed related to the activation of TGF-beta signaling in this highly metastatic cell. What we did, we isolate exosomes from MD823-1 conditioned medium, and we start to characterize, as you can observe by microscopy, these small EVs, they present the expected morphology of an extracellular vesicle. They also express typical markers such as TSG101 and ALEX, and they can activate TGF-beta signaling pathway, although we couldn't observe the activation of other signaling pathways as exemplified here by the Wnt. So it may be related to different cargo they transported when we isolated from MD823-1 cells. But one thing is to activate the TGF-beta signaling in recipient cells. But how can we better understand TGF-beta biology now with this new element that is the exosomes or these small EVs? Most studies will treat cells in vitro with increasing concentrations of exosomes or increasing number of particles. But that's complicated because these exosomes may present different concentrations of TGF-beta, and then just by concentration or by number of particles may not be exactly the best way of compare. So we ask ourselves, how can we compare the soluble ligand, the recombinant human TGF-beta that we buy, with the TGF-beta that is secreted in a small extracellular vesicle? So what we did, we treated MD823-1 cells, is we isolated all extracellular vesicles from its conditioned medium, and we added back to MD823-1 cells at increasing concentrations after establishing how much TGF-beta is transported in these small extracellular vesicles. When we evaluate this SMAD3 transcription activity, the activation of TGF-beta signaling pathway in these cell lines, treated with increasing concentrations of recombinant human TGF-beta, we can observe that, of course, increasing concentrations will increase this SMAD3 transcription activity, up to about 2 nanograms per mil. When we keep increasing the concentration, we observe a plateau and even a slight decrease in TGF-beta signaling, which is normal. This is more than expected. But this is not observed when we keep increasing the concentration of small EVs. When we have TGF-beta in small EVs, this keeps increasing the activation of TGF-beta signaling MDA221. And this is not a matter of different dynamics, different kinetics. And you can observe on the graph on the right that both treatment with recombinant human TGF-beta in gray or treatment with small EVs TGF-beta in black will have exactly the same dynamics, or very, very close dynamics over 50 hours or more in MDA221 cells. More than that, effect is not specific to MDA221. When we isolate the small EVs and we treat CEP7 cells, again, cells that are very poorly responsive to TGF-beta cells, they are very poorly migratory, very poorly metastatic cells, we can also observe an increasing response to TGF-beta in cells treated with small EVs when compared to cells treated with recombinant human TGF-beta, either by evaluating by luciferase assay or also by evaluating the phospho-SMAD2 levels by Western blot. Phospho-SMAD2 will be also higher in cells treated with small EVs compared to recombinant human TGF-beta. That's from the biochemical point of view. But activating TGF-beta should lead to functional effects. So what changes in cells treated with small EV? CEP7 cells usually grow in large patches. They are not exactly epithelial, but they still present some epithelial characteristics. And this is lost. This is expected to be lost upon treatment with TGF-beta over a few days. And we can also observe the same change in cell morphology and cell growth when we treat the cells with small EVs containing TGF-beta, the same concentration of TGF-beta. That's not only a change in the morphology of the cells, we can observe this matched change in localization of epithelial markers. Here I'm presenting an example for E-cadherin. E-cadherin is usually located at the plasma membrane of the cells, and this is lost upon treatment with TGF-beta or with small EVs. And not only the localization changes, but also the expression of these proteins change. We can observe the treatment with small EVs show a drastic reduction leads to a drastic reduction in ZO1, but also in E-cadherin. And beyond inducing an epithelial-mesenchymal transition, when we treat cancer cells, now exemplified with MDA231, with SAVs, with small extracellular vesicles, they will also induce the migration of the cells, even to higher levels than what is observed for treatment with TGF-beta. And not only migration, but also the invasion when evaluated by mitrogel-coated transwell assays. We can observe after 48 hours treatment, we can observe increased invasion of MDA231 cells treated with SVs, even compared with treatment with TGF-beta. But different of a treatment with recombinant human TGF-beta, which is very pure, treatment with SVs may contain different types of molecules. So how can we specifically determine the contribution of TGF-beta? We have different strategies in our group, either using drugs, such as SB, which is a TGF-beta receptor type 1 kinase inhibitor, or genetic by overexpressing SMAD7 and inhibiting the R-SMAD phosphorylation. Applying treatment with SB421542, and you can observe that if we treat as a control, we treat the cells with TGF-beta, they will respond and increase the SMAD3 transcription activity, but this is inhibited by treatment with SB. And the same situation happens when we treat the cells with small EVs, increasing TGF-beta signaling that is inhibited by this specific TGF-beta inhibitor. This is also observed in the migration of the cells. If we treat with, again, if we treat them with small EVs, they will increase the migration. But as we can see in the bottom right, actually in the middle of the slide, if we add this small EVs to the cell culture in the presence of this SB421542, the migration is inhibited. So we can understand that small EVs isolated from MDA231 conditioning medium can activate it, and more than activate it, When compared concentration by concentration, they can even amplify the TGF-beta signaling in breast cancer cells. But what about the physiological relevance of these small EVs? One, we can modulate and choose the concentration we want to understand the phenomenon. But the cells, they are expected to be subjected to TGF-beta, yes, but we cannot modulate in vivo in a real tumor. We cannot choose how much exosomes they are expected or they are submitted to. So to better understand the physiological relevance of these small EVs, what we did was induce the Rab27A knockdown in in MDA231 cells. So Rab27A, it's a critical protein involved in the fusion of multivesicular bars and the secretion of exosomes. So by knockdown Rab27A, we would expect to reduce exosome secretion. We can observe that the knockdown was successfully established in these cells, and that reduced at some extent the levels of ALEX and TSGO101 in the small EVs isolated from Rab27A knockdown cells. And what's more interesting is if we treat the parental MDA231 cells with increasing concentrations of TGF-beta, as expected, we will observe increasing levels of phospho-SMAD2. This also happens when we treat the Rab27A knockdown cells, but we can clearly observe that the phospho-SMAD2 levels are lower than in parental cells. So the secretion of these small EVs is indeed critical for the autocrine activation of TGF-beta signaling pathway in this highly metastatic cell. So if this is important to the activation of TGF-beta signaling pathway, this of course raises the flag that it could be pursued as a pharmacological target, and use pharmacological drugs, pharmacological agents targeting the secretion of SVs could also reduce, potentially reduce the TGF-beta signaling. There are different exosome, different drugs described in the literature that can impair the exosome docking or the docking of multivesicular bodies and the secretion of exosomes. And we choose some of them, which we tested some of these drugs. And here I'll present the results from two of these drugs, the DMA and also the vortmanine. As observed by Western blot, treatment with DMA or vortmanine will reduce the secretion of these small EVs at some extent. But more than that, when we just treat MDA231 cells with TGF-beta, they will respond to TGF-beta as shown in the black bar. But if also treat the cells with increasing concentrations of DMA, just the DMA, which wouldn't be expected in other sense to impair TGF-beta signaling pathway, will lead to a reduction in this SMAD3 transcription activity. Not only DMA, but a very similar effect is also observed for the treatment with vortmanine, with increasing concentrations of vortmanine. And what is also interesting and reinforce that this secretion of small EVs is important for highly metastatic cells, but maybe not so much for poorly metastatic cells, is that if we treat MCF7 cells, poorly metastatic cells and poorly responsive to TGF-beta with either DMA or with vortmanine, we don't have the same pattern of reduction, although we can observe at some extent with higher levels of vortmanine. So that demonstrates that DMA and or vortmanine should inhibit SV's secretion and also impair TGF-beta signaling activity in highly invasive MDA231 breast cancer cells. And if targeting the secretion of these exosomes, it's interesting to inhibit the activation of TGF-beta signaling, targeting the uptake of these small EVs should also lead to a similar result. So in addition to test drugs targeting exosome secretion, we also choose PNPXI and heparin to target exosome uptake. And very similar to what we observed before in the previous results, if we treat the cells with SAVs, they will show increased SMAD3 activity, increased TGF-beta signaling, that is reduced upon treatment with heparin, and also reduced upon treatment with PNPXI. Again, either observed by luciferase assay or by phospho-SMAD2, by Western blot evaluating phospho-SMAD2 levels. The phospho-SMAD2 level is increased upon treatment with TGF-beta, upon treatment with small EVs, and this is reduced upon treatment, co-treatment with heparin, or in particular with PNPXI. This function, this biochemical effect observed after treatment with heparin and pin-pixide. Also, it can also be observed in a functional level. MCF7 cells treated with small EVs, we show increased migration in wound healing assay, and this is dramatically reduced if we treat the cells with SVs in the presence of heparin and also in the presence of pin-pixide. So drugs targeting small EVs uptake efficiently inhibit the TGF-beta signaling activity and cancer cell migration induced otherwise by MD831 small EVs in vitro. So good drugs targeting small EVs trafficking reduce beyond in vitro the breast cancer metastasis in vivo, but also inhibiting TGF-beta signaling. Better understand this phenomenon, we're using a not-skid model mice. In this not-skid mice, non-orthotopic model, we will implant MDA231 cells. The cells are stable labeled to express the Gaussian luciferase in a constitutive manner, but it also infect the cells with the adenovirus to express the firefly luciferase in response to TGF-beta. So the cells are double labeled. In the contralateral fat pad, we will implant the wild type cells, the same MDA231 cells, but not labeled. This will allow us to do two very interesting evaluations. The labeling of these MDA231 cells with Gaussian luciferase established in our group allows the detection of metastatic cells ex vivo at a single cell level in different types of tissue. The infection with the adenovirus to express firefly luciferase in response to TGF-beta allows the evaluation of TGF-beta signaling in vivo. And the implantation of wild type MDA cells in the contralateral fat pad allows the determination of the so-called tumor self-seeding, which is very similar phenomenon to metastasis and also related to cancer progression. After implanting these cells, we separate these animals in three groups. The control group just treated with the vehicle, the saline solution, PBS, or animals treated with the small EVs secreted by MDA cells, either just treated once or treated 3 times, a continuous treatment. These animals were implanted and the treatment started on day 6, and in a relatively short experiment, animals were sacrificed or euthanized on day 20. Can observe by vivo imaging that there's not such a dramatic difference in the activation of TGF-beta signaling in animals just injected with PBS, with the control solution, or animals injected with exosomes, with small EVs. This is 7 days after the tumor implantation. When we move on to 11 days, we can observe in the graph that animals treated with the saline solution or just treated once with PBS, we're starting to show very low levels of TGF-beta signaling in vivo. And this may be a little bit higher, but still with very large variance in the group of animals treated with exosomes more than once. And this difference is starting to increase and increase when we move on to day 13th after implantation. This is still not statistically significant, but may indicate some direction to future in vivo experiments, and it's not at all unexpected. Just remember, MDA cells are very responsive to TGF-beta, so it may not be possible to observe an increasing in vivo, as these animals already possess TGF-beta and exosomes in the circulation as well. After finishing the experiment, after this 3-week experiment, we'll euthanize these animals and collect different samples to evaluate the self-seeding ex vivo by luciferase assay, evaluating the Gaussian luciferase activity. If we collect blood, we can determine the levels of circulating tumor cells, again, by luciferase assay. And if collect lungs or liver, we can detect metastasis in distant organs, again, by luciferase assay ex vivo. So we will chop these tissues, we will cut these tumors or tissues in several pieces and evaluate each of these pieces individually. Similar to what was observed for the vivo imaging of TGF-beta signaling, we could not observe any alteration, any major alteration in the metastatic levels in animals just injected with the saline solution or with exosomes over time. This is just to exemplify what we observed in lung samples also in liver samples, and the wild type tumors, which would be associated with tumor self-seeding. Again, not overall as expected, but again, may indicate some future directions of how to target the exosome secretion to reduce the activation of beta signaling. So in conclusion, exosomes or small EVs secreted by highly invasive cancer cells can amplify the activation of TGF-beta signaling pathway and increase cancer cells aggressiveness in vitro, although in vivo, we'll still have some experiments to do and some observations to make. And in overall, we want with this project in the future steps is to establish how this exosome or the small EVs can mediate in an autonomous way the activation of TGF-beta signaling pathway in tumor cells, but also the interaction between tumor cells and non-tumor cells in the tumor microenvironment. In vivo, this could lead to a very important mechanism to evaluate therapeutic agents, and overall, we would say that pharmacological inhibitors still targeting the secretion or the uptake of small EVs should still be evaluated either individually or combined, because they can potentially reduce the activation of TGF-beta signaling pathway and therefore be very important to impair breast cancer metastasis. I'd like to thank all my supervisors, Hang Jan-du and Professor Peter Dak, and all the members for the Cancer Signaling Laboratory at the University of Melbourne, the Royal Melbourne Hospital. Thank
you. Okay, thank you for this congratulations also for your presentation of your study, very interesting. I would like to start with a couple of questions. I mean, according to your in vivo data, it looks like there should be a threshold of concentration of EVs to play a role in the tumor formation. So is there any way you can quantify the secretion of microvesicle? And the other way, because it would be expected, for example, in the patients, so the tumor burden should affect the phenomenon mediated by the microvesicle.
Yeah, so it is possible to detect, and that's something that we are pursuing in our group. Some other researchers at the moment testing this. It's possible to evaluate the concentration or the number of extracellular vesicles in the circulation. This is very important, and not just by our group, but has been investigated by multiple groups, because the increased number of circulating extracellular vesicles has been proposed as an important biomarker, has been associated with cancer progression and therefore as an important biomarker, but that's in the circulation. In this sense, what I think it would be even more important, not hard to say if more important, but it's still very important, would be to determine this within the tumor, in the tumor mass. And for that purpose, this is still a bit complicated, because we need to have a separation in the method of arrangement that I couldn't see in the literature so far. So yeah, it could be done, but we're still a bit further probably to establish this as a pattern. As much as we at the moment don't know exactly, for instance, how much or what is the concentration of exosomal TGF-beta in a patient, because at the moment it is still complicated to determine just the concentration of exosomes. Even more complicated to be sensitive enough to determine how much TGF-beta we have in this exosomes in the circulation.
Okay, so we have some question here in the chat. So one is a good morning, congratulations on your presentation. And we have also assessed the concentration TGF-beta in microvesicle in addition to exosome.
Yeah, thanks for the question. That's a very, very important question. We have done this, and what we have observed is that for large vesicles, such as expected microvesicles and apoptosomes or this oncosomes, the concentration of TGF-beta, at least in the models we have tested in vitro, it's not as high in large vesicles such as in small vesicles. But that with a very important consideration when we thinking about the methodology as a very important part of this experiment. When we seed these cells to have the condition median, we want the cells to be in the best shape possible. So for in vitro experiment, this is very easy to have. And then we have the secretion of this small extracellular vesicles that we can isolate. In this scenario, the concentration of TGF-beta in small vesicles is low. Sorry, the concentration of TGF-beta in large vesicles is low. But if we simulate stressful conditions, such as hypoxia, such as overconfluence, this may change. We don't have a very careful and thoughtful study on these conditions, but we have some evidences indicating that in these stressful scenarios, maybe the concentration of TGF-beta in large vesicles may also increase.
Okay. Another question, did you study the presence of TGF-beta receptor in their types on responding cells?
Yeah, it is possible to detect the presence of TGF-beta receptors. In fact, this is one of the very interesting questions that we have asked in our group as well. Part of this biochemical amplification induced by small vesicles could be led not only by the presence of the ligand in the small vesicles, but by increasing the number of receptors in the recipient cells. That's a question that we haven't explored in depth yet, but it is possible, and there are some very interesting ways that we can investigate how the transport of TGF-beta receptors from highly responsive cells to low responsive cells would affect these low responsive cells, either genetic or pharmacologic as well. So that's a very interesting question, but we haven't this final answer yet.
Okay, a final comment that I have is, TGF-beta is certainly an important pathway and signaling in regulating the progression, EMT, and different markers. But yeah, as you might know very well, exosome or even microvesicle contain different factors, not only, are not only containing TGF. So I'm wondering if you are planning also to investigate either a pathway or signaling that can enhance even the effect of TGF-beta and can affect the metastatization in vivo.
Yeah, our group has a collaboration with another researcher here in Australia, Professor Richard Simpson. That is Professor Richard is maybe one of the most the highly regarded experts in proteomics, and was one of the very important players in the enrichment techniques to obtain more detailed results in proteomics. So my supervisor, Dr. Hongjian Zhu, and Professor Richard Simpson, they have published some papers evaluating the proteome of this small extracellular vesicles, which is very interesting, particularly interesting when they use models, EMT models, so they can compare how the composition of these small vesicles, and in this case, they have published both for small vesicles and for large vesicles, how the composition, the loading of these vesicles change when evaluating the secretion of epithelial cells from mesenchymal cells over a pole, and including a partial mesenchymal phenotype in between. And that changes, and that includes several other growth factors such as VEGF, for instance. So yeah, you may have not even a possibility, that's for sure. There are different other factors that will change the way these cancer cells behave. One important factor here is for modeling. One of the approach we take in vitro for in vitro experiments here, just to have more material, is to treat cancer cells for the isolation of small EVs. We treat these cells with TGF-beta. Very short treatments, 1 hour, just so these cells can take up the TGF-beta and secrete back, enrich these small vesicles in TGF-beta. That's for the reason of having more TGF-beta in the exosomes. So this way, it's possible that the same concentration of exosomes with lots of TGF-beta, there's not so much of other growth factors, but definitely they're still there.
All right. Okay. So thanks a lot. So we need to move forward with the next speaker. Good luck for your PhD program and your success to achieve a successful project. Thank you again. Thank you. Okay, so next speaker is Ornella Urzi from the University of Palermo, the Department of Biomedicine, Neuroscience and Advanced, sorry, I lost the diagnosis for the...
Yes, good morning to everybody.
Yes, so the title of University of Palermo, so the title of our talk is a colorectal cancer derived small extracellular vesicle induced epithelial-to-mesenchymal transition in hepatocytes, new hypothesis regarding the premetastatic niche formation in colon cancer liver metastasis. So Ornella, you can go ahead with your talk.
Thank you for the presentation, and I want to thank the organizer for selecting my abstract as an oral communication in this very interesting virtual event. It's a pleasure for me. So today I want to show you the results of our study, which aims to investigate the role of colorectal cancer-derived small extracellular vesicles in hepatic parenchyma in order to understand the mechanism underlying the premetastatic niche formation in the liver. I will be very quickly in the introduction and the background because other talks already introduce exosomes, extracellular vesicles, and epithelial-to-mesenchymal transition. But I want just to say a few words about colorectal cancer. Colorectal cancer is the third most common tumor in the world and is the second cause of cancer-related death in developed countries. More than 50% of patients affected by colorectal cancer develop liver metastasis in the course of their life, and this is the leading cause of death in these patients. In the last years, a lot of study in literature demonstrated the crucial role played by colorectal cancer-derived small extracellular vesicles in premetastatic niche priming, but especially at level of non-parenchymal cells. However, to date, there are no studies present in literature which underline the role played by hepatocytes in this step of metastasis. So the aims of our study is to study the effect induced by colorectal cancer-derived small extracellular vesicles on liver parenchyma, especially on the structural feature and functional activities of hepatocytes. Moreover, we want to study how colorectal cancer small EVs can be involved in premetastatic niche formation. First of all, I started with the isolation characterization of small extracellular vesicles from two colorectal cancer cell lines, SW480 and SW620. These are two isogenic cancer cell lines, the first isolated from the primary tumor, the second isolated from lymph node. We characterize these EVs through transmission electron microscope in order to assess the shape, the morphology of these EVs, and through Western blot in order to find the presence of CD81 and HSC70, two exosomal marker, and the absence of calnexin, a cellular marker. Then what we did was to start the treatment of hepatocytes, which are represented by THL2 cell line, with these colorectal cancer small EVs. What we observed at a gene expression level was that after 24 and 48 hours of treatment, there was a downregulation of 3 functional markers of hepatocytes, such as albumin, ApoE, and CYP3A4, compared to the control condition. These gene expression data were in agreement with protein analysis. Through ELISA, we analyzed the level of albumin released in the conditioned media of hepatocytes, and we found that following the treatment with colorectal cancer small extracellular vesicles for 24 hours, there was a downregulation and reduction of albumin released in the conditioned media compared to the control. More interesting, we analyzed through confocal analysis the expression of HNF4, which is a crucial factor in the differentiation and the maintaining of differentiation of hepatocytes. And as you can see here in green, following the treatment with both colorectal cancer small EVs, there was a downregulation of these factors. So these preliminary data lead us to think that the treatment with colorectal cancer EVs may impair the function of hepatocytes. Later, through confocal analysis, we also observed the morphology of hepatocytes treated with colorectal cancer-derived small EVs. And what we found was that following the treatment, the colorectal cancer EVs altered the morphology and the phenotype of hepatocytes. The cells lost contact with each other and started to assume a mesenchymal-like morphology. Since TGF-beta1 plays an important role in colorectal cancer progression and metastasis, and is finally the master regulator of epithelial to mesenchymal transition, we thought to investigate its presence in colorectal cancer-derived small EVs. We analyzed TGF-beta1 presence in small extracellular vesicles through Western blot, and to confirm this data, so the presence of TGF-beta1 in vesicles, we also performed an analysis with an high-resolution microscope. Thanks to this, we confirmed the localization of TGF-beta1 into our small EVs. Moreover, we found that TGF-beta, that is represented in red, co-localized with two exosomal markers such as CD63 and CD9 in yellow and in blue respectively. Moreover, we test the presence of the receptor of TGF-beta into our target cells, and as you can see, hepatocytes express TGF-beta receptor 1. So we studied also the activation of TGF-beta pathway, in particular the canonical pathway, so we studied SMAD, and the non-canonical pathway, so AKT1. And what we found was that after 1 hour treatment with both colorectal cancer small extracellular vesicles, there was an upregulation of the phosphorylation of SMAD levels and a phosphorylation of phospho-AKT compared to the control conditions. So we concluded that our target cells were able to respond to TGF-beta present into small extracellular vesicles. TGF-beta is known to induce epithelial-to-mesenchymal transition through the activation of several genes, such as snail and slug. For this reason, we decided to study the expression of these two actors into our target cells following the treatment with colorectal cancer small extracellular vesicles. What we found was that already after 6 hours of treatment, target cells increased the expression of snail levels compared to the control condition, while after 12 hours of treatment, we observed an upregulation of slug levels. Epithelial to mesenchymal transition is a process in which epithelial cells lost their cell-cell contact and gain migratory properties in order to transform themselves into mesenchymal cells. So we decided to study two epithelial markers such as cytokeratin 8/18 and hicaderin. What we found was that already after 24 hours treatment, the expression of cytokeratin 8/18 was strongly downregulated compared to the control, and this was confirmed and maintained after 48 hours of treatment, when we also observed a reduction of hicaderin. On the other hand, we also studied the expression of two mesenchymal markers, such as vimentin and alpha-smooth muscular actin. What we found was that levels of vimentin were increased already after 6 hours of treatment, and this is in agreement with data in literature which describe vimentin as an early gene of epithelial-to-mesenchymal transition. The upregulation of vimentin was also maintained after 24 and 48 hours of treatment, when we observed also an upregulation of alpha-smooth muscular actin, especially by small extracellular vesicles isolated from SW620. In order to confirm the involvement of TGF-beta1 in colorectal cancer small extracellular vesicles mediated epithelial-to-mesenchymal transition in hepatocytes, we introduced asparagine into our experiment. Asparagine is a natural compound which is able to inhibit TGF-beta1. What we did was eco-treatment of hepatocytes with asparagine and small extracellular vesicles, and we found that the eco-treatment was able to attenuate the treatment with vesicles alone. As you can see here for vimentin, after 6 hours treatment, we saw an upregulation, as I showed you before, but when the cells were co-treated with the small extracellular vesicles and asparagine, this upregulation was attenuated. And we can see the same after 48 hours of treatment. When we treat the cells with small extracellular vesicles from SW620 alone, there was an upregulation of alpha-smooth muscular actin and vimentin, but the co-treatment attenuated this upregulation. Finally, in order to confirm all our in vitro data, we isolate extracellular vesicles also from the plasma of patients affected by colorectal cancer. First of all, we performed a Western blot analysis in order to ensure the purity of our samples. We analyzed the presence of CD81 and helix in our samples and the absence of calnexin. Moreover, we analyzed again the presence of TGF-beta1 that was present in almost all the samples of colorectal cancer patients' plasma EVs, even if at different levels. Moreover, we treat hepatocytes with this EVs, and we found that after 6 hours treatment of hepatocytes with EVs derived from the plasma of patients affected by colorectal cancer, there was an upregulation of SNAI levels of SLAG and of vimentin already after 6 hours, while after 48 hours of treatment, we saw an upregulation of alpha-smooth muscular actin and of vimentin again. And these data were also improved by the fact that when we treated hepatocytes with small extracellular vesicle derived from the plasma of healthy donors, no effect were observed. Finally, thanks to a collaboration with the University of Gothenburg, we also obtained small extracellular vesicles from tissue biopsy of colorectal cancer, and we performed a confocal analysis in order to analyze the expression of cytokeratin 818 as an epithelial marker and vimentin as a mesenchymal marker. After 24 hours of treatment with colorectal cancer biopsy, small extracellular vesicles, we observed surely a difference on the shape and morphology of hepatocytes, an impairment of the expression of cytokeratin 818, and an increase of the red signal, which represents vimentin, compared to the control condition and compared to the cells which received EVs isolated from healthy mucosa biopsy, thus confirming our in vitro data. So to conclude, here I show you our proposed model in which cells from the primary tumor release small extracellular vesicles, and these vesicles go to the metastatic site, so the liver. In the liver, these vesicles are uptaken by hepatocytes, which undergo into an epithelial-to-mesenchymal transition. So these cells start to lose their epithelial marker, gain some mesenchymal markers, and contribute to the liver fibrotic environment, thus contributing to the formation of the metastatic niche. I want to thank all the special people that work with me every day, especially the people that contributed to this great work. And I want to thank all of you for your attention.
Okay, thank you for your talk, interesting data. I would like, yes, to start with some questions while we wait for other attendees to post their question. I'm wondering if you had the chance to compare the release of microvesicle exosome from cell lines, I mean colorectal cell lines with different, for example, from primary as compared to metastatic, in order to compare the content, and the same as from the plasma of patients that you have utilized and characterized for the content of TGF-beta. Did you have a chance to analyze, for example, if there was any correlation from the content of, in particular, TGF-beta or extravesical, depending, for example, the site of the colon, because there are also a difference from the colon rectal cancer arising in the left compared to right side of colon, if it's a rectal cancer, or for example, even patient with a hepatic metastasis.
Thank you for the question. Till now, I worked with just these two cell lines, so SW480, SW620. These are both not metastatic cell line, even if SW620 are more aggressive. The fact is that we want to study the effect on the metastatic sites. We want to study the exosomes, the extracellular vesicles isolated from a primary tumor. As for the patients, in fact, we selected patients with colorectal cancer, so the same kind of cancer for all the patients without metastasis, because the aim, the hypothesis is that already at the beginning, when there is just the primary tumors, these cells from the primary tumors release V's that in some way come to the liver thanks to the bloodstream, and there, thanks to TGF-beta, induce the priming for the premetastatic niche. It could be interesting, of course, to compare the content of EVs, for example, doing a proteomic analysis, but we are studying also the RNA content of these EVs, because surely some difference could be interesting to analyze to understand better the mechanism of premetastatic niche formation. Thank you for the question.
Okay, thank you. Yes, I mean, I agree, but I mean, I think still it should be interesting to characterize, I mean, the microvesicle and among, I mean, even patients with the primary tumor, same primary tumor, you know, there are different types of colorectal cancer. Some are microsatellite stable, some are more stable. So try to associate with these features with the characteristic of the type of tumor of patients. We have also other question here in the chat, so congratulations. Would that be possible to knock out TGF-beta1 secretory cancer cells in order to understand its proportional impact on hepatocytes?
Okay, we don't have the possibility to do this in lab, or we are not think to do this till now, because I show you, we just used an inhibitor just to confirm the implication of TGF-beta, but of course it could be interesting to do a knockout. But anyway, as we said before in the previous talk, I think that knockout the TGF-beta, it could be nice, it could be good, okay, but anyway, extracellular vesicles contain a lot of molecules, of biological molecules, which could promote the epithelial-to-mesenchymal transition into hepatocytes. So I think that blocking TGF-beta 1 could be interesting, could be useful, but that this could just not stop the whole process of epithelial-to-mesenchymal transition.
Okay, so and then a continuation from the previous question is if you or your group as explorer is a secretory extravesicle secreted by CRC in association with inactivating mutation in the TGF-beta signaling component, and if you would expect any difference in their impact on hepatocytes.
Okay, we didn't try to do this, but it could be interesting because we saw that hepatocytes possess the receptor for TGF-beta, so some mutation in TGF-beta 1 on the extracellular vesicles could impact to the response to this signal. So it could be really interesting to do that, so thanks for the suggestion. And we could think to do this kind of experiment. Not yet. We didn't not yet, but we could think to do this. Thank you so much.
Okay, I don't see any other question, just a thank you note for your answer. And so thank you for your talk and congratulations again. So we can move to the next speaker. The speaker is Didem Caracas-Zeybek from molecular biology. Okay, always. Sorry, because when you open the presentation, my electronic, okay, disappear. Okay, so I was saying from the Department of Molecular Biology and Genetics Faculty of Engineering and Natural Sciences from Turkey. So the title of the talk is the role of exosomal communication between pancreatic cancer cells and Schwann cells on the aggressiveness of pancreatic cancer. So please, the stage is yours.
So good morning, everyone. I'm Didem and currently working at the Department of Molecular Biology and Genetics in İstinye University, Turkey. And today I'm here to present a part of my recent research. And before I start my talk, I would like to thank the organizers to provide me this opportunity. And I'm really happy to be here, by the way. And today I will briefly talk about the possible or potential roles of exosomes in the interaction between pancreatic cancer cells and the cells in nerve structure, especially Schwann cells. So as we know, pancreatic cancer is one of the most deadliest cancers among all cancer types. Despite all the efforts, the survival rates of the patients remain almost similar by year. And in 2021, approximately 60,000 people will be diagnosed with pancreatic cancer, and unfortunately, around 80% of them will die until the end of the year. So when the patients have diagnosed with pancreatic cancer, they have already, more than half of them have already metastasis. And the metastasis drastically reduces the 5-year survival rate of the patients. So we traditionally can mention 3 main routes for the metastasis of the pancreatic cancer. So the cells may spread via blood circulation, or they may use lymphatic system, or they may spread in peritoneum. But in addition to these traditional ways, We also know that cancer cells, especially pancreatic cancer cells, can also invade the nerve structure. So when these cells invade the nerve structure, they may grow and proliferate in three main layers of nerve, which are epinerium, perinerium, and endonerium. That's why actually the invasion of cancer cells to nerve structure is generally called neural invasion. But we also know that most cancer cells which invade the nerve structure dominantly grow in perinerium. And that's why the process is also known as for perineural invasion, which is more common terminology to define this process. So basically, perineural invasion is a process of invasion of the cancer cells to nerves, and which is defined as under-recognized route of the metastatic spread. When we look at the structure of normal pancreas, we know that pancreas is well innervated organ, and there are lots of nerves arise from especially circular ganglia, and this this nerve-rich environment somehow provide as an alternative or easily accessible route for the cancer cells, which means that this nerve-rich environment helps cancer cells to perineural invasion process. And as you can guess, compared to other cancer types, pancreatic cancer have the highest incidence for the perineural invasion, and the patients who have perineural invasion are correlated with poor prognosis and increased pain. In addition to perineural invasion, the invasion of cancer cells to nerve structure, we also know that compared to normal pancreas, in tumor tissues, the density and the size of nerves is also changing. And the survival data show us the patients who are nerve positive have shorter overall survival rate compared to the patients who nerve negative. And we can say the same thing about the size of the nerve as well. So the patient's bigger nerve causes shorter survival rate in pancreatic cancer patients. So here the point is that so far what we know, okay, the cancer cells may invade the nerve tissues, and similarly, they also manipulate the nerves around themselves. So they increase their density and they increase the size of the neurons, which means that somehow the cancer cells may also manipulate the nerve structure. And in addition, some studies show that some neurons, some nerve structure infiltrate inside the tumor as well, which means that there is a kind of bidirectional interaction between cancer cells and the cells which are found in nerve structure. So far, the mechanism of the interaction between nerve cells and cancer cells is unclear, but quite recent studies highlight the importance of Schwann cells in this communication. So when the cancer cells stimulate the Schwann cells by releasing some factors, they cause dedifferentiation of the Schwann cells. As you know, normally the function of the Schwann cells to cover neurons. So when they're stimulated by tumor cells, they are dedifferentiated. And after this point, they start to promote the aggressiveness of the cancer. Then somehow these cells guide cancer cells and help them. And with the help of the Schwann cells, the cancer cells can reach to neuron structure, which means that the Schwann cells helps cancer cells for the perineural invasion process. The mechanism, so far some factors have been defined in the interaction between nerve cells, which might be neurons or Schwann cells, and pancreatic cancer cells. So far, we know that some neurotrophic factors, chemokines, and neurotransmitters may involve in the interaction between cancer cells and the nerve cells. But the studies about the role of exosomes in this communication are really limited. That's why in this study, we focused the exosomal interaction between pancreatic cancer cells and neurons, and pancreatic cancer cells and Schwann cells, so two main cell types in nerve structure. First of all, we isolated the exosomes from pancreatic cancer cells and treated neurons with these exosomes, and we analyzed some changes in neurons. Then we also treated pancreatic cancer cells with the exosomes derived from neurons and analyzed the aggressiveness of the pancreatic cancer cells. And we did exactly the same thing for the interaction between Schwann cells and pancreatic cancer cells as well. So let's take a look at our results. First of all, we differentiated SHSY5Y neuroblastoma cells to neurons by treating them with all trans-retinoic acid. And we just named these cells as SHSY5Y-N cells, which indicates neuronal cells. Then we confirm the efficiency of this differentiation by Western blot. So here you can see NT indicates non-differentiated neuroblastoma cells, and these are differentiated cells. So basically here we tried 3 different cell culture formula to select the best differentiation formula for the cells. And at the end of the experiment, considering the changes in the neuronal markers like neuron-specific enolase or beta-3-tubulin or neuronal nucleus marker, as you can see here, compared to non-treated group, these markers are increased, which indicates the efficiency of our differentiation. So we just select the third condition, and then we continued to our experiments with this condition. So we also confirmed the differentiation by some morphological analysis as well. Here, this picture list shows undifferentiated cells, which means that these are neuroblastoma cells. So in culture, these cells tend to grow little bit cluster-like structure, and there's not much extensions like small branches between the cells. I hope you can see, but when we differentiate these cells into neurons, here you can see the cell's morphology is changing a little bit, and I'm gonna show some of them. Here you can maybe see some tiny branches between the cells, which are called neurid structure. So which indicates, actually, which is a kind of morphological marker for the neurons. And we also stain these neurids by using a specific antibody beta-3-tubulin, and this, and as you can compare to non-treated or non-differentiated neuroblastoma cells, when we differentiated the cells, here you can see the tiny branches, neurids between the cells. So since we use exosomes in all of our experiments, we also characterize the exosomes by Western blot by using some specific exosomal markers, CD60, CD81, and CD9. And we also show the uptake of the exosomes by cells by staining the exosomes with specific marker. And here you can see the uptake of the exosomes by the cells. So in the next step, we isolated the exosomes from pancreatic cancer cells and then treated the neuron with these exosomes. By the way, here we use 2 different types of pancreatic cancer cells, which are PANC1 and BXPC3 cells. And then we check the changes in the migration ability of the neurons. Non-treated indicates there is no exosome treatment, and here we treat the cells with PANC1 and BXPC3 exosomes, as you can see here. And when we quantify our results, while PANC1 exosomes slightly induce the migration of neurons, but it's not statistically significant, BXPC3 induced the migration of neuron cells. Then we also analyze the changes in the density of neurid structure of the neurons when they exposed to exosomes. And here you can see, compared to non-treated group, PANC1 exosome and BXPC3 exosomes increase the neurid density in these cells. We also analyze the changes in some neuronal markers, neurospecific analyze and beta-3 tubulin, but compared to non-treated group, we couldn't find any significant changes in the expression of these proteins. So, since we know there is a kind of bidirectional interaction between cancer cells and neurons, in the next step, we isolated the exosomes from neurons and then gave them to cancer cells. Then we checked some, we performed some functional tests to analyze the effects of the exosomes on the aggressiveness of the pancreatic cancer. Briefly, here you can see PANC1 and BXPC3 cells, which are treated with neural exosomes, this line and that line. And when we quantify the migration, our migration result, there is no statistically significant changes in the migration ability of PANC1 cells and BXPC3 cells when they exposed to neuron-derived exosomes. And actually, in PANC1 cells, we may say there is a kind of slightly increase, but this is not significant, statistically significant. And we got almost the same pattern for the invasion assay. And when we treated the cells with exosomes, the exosomes slightly induced invasion of the cells. And in BXPC3, there is no significant changes in the cell invasion. Then we also, since we couldn't get promising results with BXPC3 cells, we also performed colony forming assay with by using PANC1 cells only. When we quantified the result, we couldn't find any significant changes in the colony forming ability of the cells. We also performed cell viability assay to assess the effects of exosomes on the viable proliferation ability of the cancer cells. I didn't include the figure here, but we couldn't find any significant difference as well. So since we couldn't get much promising result with cancer cells and neurons, we just changed the focus of our study. And after that point, we focused the interaction between cancer cells and the Schwann cells. Because as we know, the Schwann cells covered the nerve structure and they are accepted as the first contact point in the interaction between neuron and cancer cells, which means that first cancer cells and Schwann cells need to interact each other. Then this interaction will affect the neuron and cancer interaction. So by using this information, first of all, we isolated the exosomes from cancer cells and treated Schwann cells with the exosomes and analyzed some further experiments. We performed some further experiments. So while the PANC1 cells derived exosomes didn't change the cell proliferation in Schwann cells, they significantly increased the migration of the cells. By the way, we performed all the experiment by using PANC1 and BXPC3 cells-derived exosomes, but we got the most promising result with PANC1 cells exosomes. That's why, after that point, we focused the interaction between Schwann cells and PANC1 cells. So, consistent with two-dimensional migration data, the cell migration was also found to increase in three-dimensional environment as well in the presence of the exosomes. And similarly, the morphology of the Schwann cells looks a little bit changed, that the cells look more spindle-like structure in the presence of PANC1 cells derived exosomes. And consistent with these data, PANC1 cells derived exosomes increased invasion ability of Schwann cells. And the most exciting thing, when we treated Schwann cells with PANC1 cells exo, we found that the expression of glial fibrillar associated protein, or shortly GFAP, which is a marker for dedifferentiated Schwann cells and which indicates the presence of tumor supportive Schwann cells. So we found that GFAP expression dramatically increased when we treat the cells with the exosomes derived from PANC1 cells, which means that somehow the exosomes, PANC1 cells exosomes induce the differentiation of the Schwann cells. So in the next step, of course, we also wanted to analyze the effects of Schwann cells on the PANC1 cells, the effects of the exosomes on PANC1 cells. So for this purpose, first of all, we isolated the exosomes from Schwann cells and then treated PANC1 cells with the exosomes. But unfortunately, we couldn't get any significant result. Then just we criticize the literature and discuss what we know about the interaction between the cancer cells and the Schwann cells. And then we realized that in normal tissue, first, the Schwann cells are stimulated by cancer cells. Then they are differentiated and they have tumor supportive phenotype. So to briefly or to simply mimic this tissue-like environment, here we just treated Schwann cells with the conditioned media, which we collected from PANC1 cells. So we treated Schwann cells with PANC1 cells derived conditioned media for 24 hours. And then we changed the media and kept the cells in fresh media for an additional 24 hours. Then we isolated the exosomes from these cells, cancer-stimulated Schwann cells. And then we gave the exosomes to PANC1 cells. Then, of course, we analyze the changes in the cells which are related to aggressiveness of the cells, especially metastatic capability of the cell. First of all, when we treated PANC1 cells with Schwann exosome, the cell viability significantly increased, or cell proliferation significantly increased in both 24 and 48 hours. And consistent with this data, cell migration is also increased compared to non-treated group when we treat PANC1 cells with Schwann cells exosomes. And similarly, cell migration increased, and three-dimensional cell migration was also found to increase. I know it's not clear, but here the matrigel is, the line of the matrigel is almost disappear, and actually there are lots of cells which evade from matrigel. And while there's no changes in the colony forming ability of the PANC1 cells when they treated with Schwann exosomes, we found some changes in the expression of proteins which are related to epithelial-mesenchymal transition of the cancer cells, especially when we treated PANC1 cells with exosome, vimentin, SLUG, and encoderine levels dramatically increased. And consistent with the protein data, we also observed that when we treated PANC1 cells with Schwann exosome, cell morphology become more mesenchymal. So cells have more mesenchymal-like structure compared to non-treated group. So after that point, we wanted to make our study a little bit more specific. And here And as we know, the exosomes contain lots of components like DNA, RNA, protein. And here we just decided to focus the role of microRNAs in these exosomes. So for this purpose, we isolated the exosomes from PANC1 cells and SHI1 cells, and then analyzed the exosomes for the expression of 84 different microRNAs by using PCR array. And then we focus SHI1 cells exosomal part, because there's not much information in literature about the exosomal content of the SHI1 cells. So when we look at the SHI1 cells exosomal microRNA profile, some microRNAs are overexpressed, as you can see, and some of these overexpressed microRNAs are tumor promotive, they have tumor promotive function, and some of them are oncogenic. That's why actually, considering this information, we selected microRNA 125B5P as a potential candidate, because which is overexpressed in SHI1 cells, exosomes, and more than twofold. And when we search something about the literature, it is an oncogenic mirror. So that's why actually we just focus the function of these microRNA. Now we are trying to confirm the potential role the microRNA 125B in PANC1 cells. So briefly, we just overexpressed and suppressed this microRNA in these cells by using microRNA mimics and inhibitors, and then evaluated the changes in cell migration and invasion. So this part is still ongoing. So that's why actually we can only perform the experiment only one time, and we will just arrange the experiment condition and then just repeat the experiment a few times, of course. So this part is still, that's why ongoing. So as a result, we can say that when we treated neurons with pancreatic cancer-derived exosomes, cell migration and neuron density is increased. And similarly, when we gave the exosomes derived from neurons to pancreatic cancer, there's not much changes in the migration and the invasion of the cells. The most exciting part was the interaction between SHI1 and cancer cells for us, because when we treated SHI1 cells with pancreatic cancer-derived exosomes, cell migration invasion are increased, and we also show that the differentiation marker GFAP expression significantly increased in these cells. And similarly, the exosomes derived from SHI1 cells is also increased migration, invasion, proliferation, and EMT-related markers in PANC1 cells. So our future plan is to analyze the role of microRNA 125B5P on PANC1 cells aggressiveness. And as a future, little bit later future plan, we also want to evaluate the role of SHI1 cell-derived exosomes in the communication between pancreatic cancer cells and the neurons. So thank you for your attention. And this is a photo from our research group. And I would like to especially thank to our group leader, Dr. Ulukaya, and some of my colleagues, especially Naz, Merve, and Okan, for their supports. And if you have any question, I would like to be happy to answer them. Thank you.
Okay, so thanks a lot. Very interesting and promising study. We have a question here in the chat, so congratulations, Didem, great talk. I'm wondering what is the role of exosome for the interaction of these cells within the tumor microenvironment? Could they establish direct cell-to-cell contact? And have you or your group tested the impact of Packer cells on Schwann cells or vice versa using co-cultures?
Okay, thank you for the question. That was really interesting and nice question. So actually, first I will answer the first part. And in tumor microenvironment, we know that the exosomes help to communicate in the cells, especially in long distance. And so the cells derived from especially cancer cells manipulate the microenvironmental cells, and then these cells become to promote the cancer aggressiveness. But so far, we don't have much idea about the role, the role of the exosomes derived from neural cells or the Schwann cells. As far as I know, there's only one study has been published which shows the role, which shows the exosomal communication between cancer cells and the neural cells, as far as I know. But it's really, unclear. That's why we focus to understand the role of these cells. And another question is actually, from, as far as we know from the literature, they establish direct cell-to-cell contact, and this cell-to-cell contact help. When the cancer cells and Schwann cells contact each other, which increase aggressiveness of cancer cells. We know this information from the previous publications. And here, I haven't tested direct co-culture experiment, but I performed indirect co-culture experiment by using Schwann cells and cancer cells. But to be honest, I did the experiment only one time, and since the Schwann cells are really proliferative, I needed to optimize probably the experiment condition, but we are also planning to work on it. And by the way, we also analyze the effects of conditioned media derived from these cells on, pancreatic cancer cells or Schwann cells as well. And we also get slightly increases when we use conditioned media as well.
Okay, very good, very interesting. Actually, I have a curiosity because, the pancreatic cancer are very resistant to immunotherapy, and they hide themselves also from, their susceptibility to immune responses is very low. They are resistant. So I'm wondering if you had chance or you're planning to do any, to characterize if the exosome, the microvesicle released by Schwann cells can in some way affect the immunological profile of the pancreatic cells.
Yeah, that is really nice question. Actually, one of my other ongoing projects, we are working on how can I say it? We are trying to modify the dense stroma and of pancreatic cancer cells, and by doing that, we are planning to increase the infiltration of natural killer cells in the tumor structure. And I sometimes actually am just, thinking about what if I combine these two experiments? So maybe I can use the exosome derived from these cells and then maybe try their effects on this system. It would be really interesting. But now I'm just still to focus to complete these experiments. Then maybe, as a next step, we can just evaluate it, which will be really, which will provide really helpful information, because these cells, the importance of these cells are gaining, and the scientists start to focus the function of the nerve cells and the Schwann cells in microenvironment. So somehow they may affect the interaction between cancer cells and the immune cells as well.
Okay, yes, it would be very interesting, this. And we have actually another question here. Thank you for your answer. So it's the continuation from the previous question. If using co-culture, the genetic inhibition of exosome secretion, for example, with RAB5 or RAB27A knockdown, or drug treatment, for instance, with the GW4869, could help to establish the physiological role of exosome in mediating the interaction between the cells, exosome, and cell-to-cell contact. So this is just a sort of suggestion or comment.
Yeah, thank you for your suggestion. It's really valuable for me. Yeah, thank you so much.
And here is another question. Since Schwann cells, they differentiate, is there any influence of myelin production?
To be honest, we haven't checked it, but as far as we know from the literature, when they are differentiated, they lost their myelinating function, and they start to release some growth factors and chemokines, and their function is just to support the promotion of the tumor cells. By the way, when the neurons are injured, the Schwann cells are also dedifferentiated to restore the damage on the nerve structure. And it's the same. So when they're dedifferentiated, they start to produce loss of growth factor, as far as I know.
Okay, thanks a lot. Thank you.
Thank you so much.
So we are having a coffee break now. Since we are slightly late in